ಎಲ್ಲರಿಗೂ ನಮಸ್ಕಾರ. ಎಜುಕೇಷನ್ ಆನ್ ಕ್ಲೌಡ್ಸ್ಗೆ ಸ್ವಾಗತ. ನನ್ನ ಹೆಸರು ನಿಮಿಲಾ. ಸೊ ಇವತ್ತಿನ ಕ್ಲಾಸ್ನಲ್ಲಿ ನಾವು ಸೆಕೆಂಡ್ ಪಿ ಯು ಸಿನ ನೆಕ್ಸ್ಟ್ ಚಾಪ್ಟರ್ ಆದಂಥ ಬಯೋಟೆಕ್ನಾಲಜಿ ಪ್ರಿನ್ಸಿಪಲ್ ಆ್ಯಂಡ್ ಪ್ರೊಸೆಸಸ್ನ ಕೆಲವೊಂದು ಇಂಪಾರ್ಟೆಂಟ್ ಪಾಯಿಂಟ್ಸನ್ನು ನೋಡೋಣ. So firstly, what is biotechnology? So it refers to the technology which is using biology. Andre, it is the applications in agriculture, food processing industries, medicine diagnostics and bioremediation and waste treatment and energy production. So biotechnology is a science of biology wherein it is used in all these strings for improved methods. Now the European Federation of Biotechnology EFB will define biotechnology as integration of natural science and organisms, cells, parts thereof and molecular analogous for products and services. Now coming to the basis of modern technology. So firstly it is genetic engineering, wherein it is the introduction of the foreign genetic material, which is DNA and RNA into the host's genome and altering the phenotype of it. Phenotype and the physical state of it. Now next is the aseptic techniques. So what are these? It involves the maintenance of contamination free ambience. Andre ಒಂದು contamination illade chemical engineering processes na ಈ ಮ್ಯಾನುಫ್ಯಾಕ್ಚರಿಂಗ್ ಆಫ್ ಪ್ರಾಡಕ್ಟ್ಸಿಗೆ ಯೂಸ್ ಮಾಡೋದು. ಫಾರ್ ಎಕ್ಸಾಂಪಲ್ ಆಂಟಿಬಯೋಟಿಕ್ಸ್, ವ್ಯಾಕ್ಸಿನ್ಸ್ ಎಕ್ಸೆಟ್ರಾ. ನೌ this is done so as to enable the growth of only desired microbes which are responsible for a bioprocess which is required. So ಈ aseptic procedures ಅಂದರೆ ಒಂದು sterilized environment wherein it is created to produce only the necessary microbes. Now coming to genetic engineering, it is the asexual reproduction which preserves the genetic information, while sexual reproduction will preserve variations. So now how are these studies? Plant and animal hybridization procedures will often result in the introduction of the undesirable genes along with the desirable ones. Now genetic engineering will overcome this limitation. So how will it include? It includes the creation of recombinant DNA, gene cloning, gene transfer into the host organism, introduce piece of DNA which does not replicate in the host unless it is integrated with the chromosome of the host. And then for getting replicated, the foreign DNA must be integrated into the host DNA sequence which has the origin of replication. And when the integration is going to occur, the foreign DNA is therefore replicated into many copies and therefore many copies of the DNAs are formed. And this is called the cloning procedure, wherein it is a process of formation of identical copies of the same DNA. Andre ಒಂದೇ ರೀತಿಯ DNA multiple copies ಈ ರೀತಿ. Now construction of recombinant DNA, which is plasmid. It is autonomously replicating circular extra chromosomal DNA which is isolated, and plasmid DNA will act as a vector because it is used to transfer the piece of DNA which is attached into the host. Now plasmid DNA is also consisting of genes which are responsible for providing the antibiotic resistance to the bacteria. And this plasmid DNA is cut at specific restriction enzyme site which is REN, and then with the help of molecular scissor which is they cut a DNA at specific location. So these are the molecular scissors wherein they cut at the DNA specific locational site. Now DNA of interest which is to be inserted is also cut with the same restriction enzyme, and now the DNA of insert is hybridized with the plasmid with the help of the DNA ligase. DNA ligase is the one which is going to stick it to form a recombinant DNA, and this recombinant DNA is then transferred to the host like E. coli, wherein it is going to replicate by using the host's replication machinery. Now E. coli is cultured in a medium which consists of antibiotic, and only cells containing the recombinant DNA will be able to survive because of the antibiotic resistant genes, and one will be able to isolate the recombinants too. Now restriction enzymes as tools of RDT, which is recombinant technology. Now restriction enzymes are the specialized enzymes which are going to recognize and cut a particular sequence of DNA. So DNA na ಇಷ್ಟು ಉದ್ದದ DNA ನಲ್ಲಿ we need one particular sequence. So these restriction enzymes are the ones which are going to help us cut the DNA at the specific site. Now nucleases, which are of two types. One, endonucleases, which will cut DNA at specific positions within the DNA, and exonuclease will cut the DNA at their ends, either 5' or 3', and they'll remove the nucleotides at the ends of the DNA. Now every restriction enzyme will identify different sequences, which is the recognition site. So identification site and now recognition site antivi. And over 900 restriction enzymes have been isolated so far, and all of which they recognize different sequences. And recognition sequences are palindromics, and which are the palindromes, and they are the sequences of base pairs which will read same on both backwards and forwards. Andre it is 5', 3' in any of the direction. So ಇಲ್ಲಿ ನಾವು ನೋಡಿದಾಗ ಈ 5', 3' ಎಂಡ್ ನೋಡಿದಾಗ GAATTC ಇರುತ್ತೆ. ಅದೇ ರೀತಿ 5' ಇಂದ 3' ನೋಡಿದಾಗಲೂ GAATTC ಇರುತ್ತೆ. ಸೊ ಆಸ್ ಅ ರಿಸಲ್ಟ್ ಓವರ್ ಹ್ಯಾಂಗ್ಸ್ ಅಂದರೆ ಸ್ಟಿಕ್ಕಿ ಎಂಡ್ಸ್ ಆರ್ ಜನರೇಟೆಡ್ ಆನ್ ಈಚ್ ಸ್ಟ್ರ್ಯಾಂಡ್. ಸೊ ಇಲ್ಲಿ ಏನಾಗುತ್ತೆ, ಪ್ರತಿಯೊಂದು ಸ್ಟ್ರ್ಯಾಂಡಿನ ಮೇಲೂ ಒಂದು ಸ್ಟಿಕ್ಕಿ ಎಂಡ್ ಬರುತ್ತೆ. Now sticky ends will form hydrogen bonds with their complementary counterparts with the help of the DNA ligases. So all of these processes will form the basis of RDT respectively. So this is the diagrammatic representation of the sticky end, as you can see here. Now naming the restriction enzyme. So first letter, genus of the organism from which the enzyme is derived is represented as first letter. Second and third letter will be the species of the organism. Fourth letter will be the name of the strain. And Roman number will be the order of isolation. And if you take an example in Eco RI, derived from E. coli, strain R, it is the first to be discovered. Now coming to gel electrophoresis. What is this electrophoresis procedure? So the fragments obtained after cutting with the restriction enzymes will be separated using gel electrophoresis technique. So now what is done here? Electric field is implied to the electrophoresis matrix, which is the agarose gel in 90% of the cases, and negatively charged DNA fragments will move towards the anode. So there will be wells which will be dug. Okay, so this is the electrophoresis matrix which is made with the help of agarose gel. So agarose gel thick ಆಗಿರುತ್ತೆ ಸ್ವಲ್ಪ, and the wells will be made, and here the DNA will be inserted, and from the cathode, when the electric field is applied, it is going to move towards the anode, and they will create bands. So now fragments will separate according to their size by sieving properties of the agarose gel, and smaller the fragment, farther it moves. So the small fragments will move faster and they will move far. And staining dyes like the ethidium bromide followed by the exposure of UV radiations are used to visualize the DNA fragments. So this ethidium bromide is very dangerous to be inhaled. It will give bluish appearance under UV radiations. And DNA fragments are visible as bright orange colored bands in this agarose matrix. And these bands will be further cut in the agarose gel and extracted from the gel piece, which is the elution. And DNA fragments which are purified, and these purified DNA fragments will be used for constructing the recombinant DNAs respectively. Now coming to the vectors. So to obtain these recombinant DNAs, we need certain vectors and host as tools of RDTs. So now coming to the cloning vectors. So plasmids and bacteriophages are commonly used as the cloning vectors, like BAC, which is the bacterial artificial chromosomal vector, and YAC, and there are so many. And both of these will have the ability to replicate within the bacterial cells and independent of the chromosomal DNA. Now bacteriophages ಗೆ ಬಂದಾಗ they have high copy number of genomes within the bacterial cell, and plasmids ಗೆ ಬಂದಾಗ it might have only 1 or 2 copies numbers to produce 15 to 100 copies per cell. Now if foreign DNA is linked to these vectors, it is multiplied to the number equal to the copy number of the vector. And now features which are present in the vector itself will help in the easy isolation of the recombinants from non-recombinants. So this is a cloning vector, wherein you can see this is the Eco RI, PUV1, PST1, and these are the chemicals. Okay, this is the PBR322 vector. Now origin of replication, ORI. So replication will start from ORI. Any fragment of DNA when linked to the ORI can be made to replicate. And with the help of this, the genetic engineer may control the copy number of the recombinant DNA. And to recover a high number, suitable origin of replication is to be chosen. Now selectable marker. So these genes will help to select recombinants over, and then they'll help as non-recombinants, and antibiotic resistant genes such as AMP-R, which is the ampicillin resistant, and TET-R, which is tetracycline resistant, will serve as selectable markers usually. So in the circle where it was measured as TET-R and AMP-R, it was the selectable marker. Now coming to cloning sites, they refer to the recognition site for restriction enzymes like EcoRI, HIND3, PUVL and BAM-HI, etc. Now these are the sites where restriction enzymes will cut the DNA. And cloning process will become completed when more than one recognition sites are present, and therefore ligation is carried out only at the restriction sites present on the antibiotic resistant genes. Now how are these antibiotic resistant genes helped in, how do they help in selecting the recombinants andre? So suppose TET-R, which is tetracycline R gene, has BAM-H1 recognition site. When BAM-H1 is used for restriction, foreign DNA fragment is inserted with the TET-R gene. And hence tetracycline resistance is not present in the recombinants. So recombinants will grow on the media which consists of ampicillin, but will die on the media which consists of tetracycline. On the other hand, non-recombinants will grow on the medium containing ampicillin as well as on the medium containing tetracycline too. In this way, this antibiotic resistant gene will help in the selection of transformants. So it will help us know which are the transformants. Now alternate selectable markers. Other than antibiotic resistant genes, alternate markers can also be used. So one of them is gene coding for galactosidase. So when foreign gene is inserted within a galactosidase gene, the enzyme A-galactosidase will get inactivated, which is insertional inactivation. So this process is called insertional inactivation, and then bacteria are grown on the chromogenic substrates, and non-recombinants will produce blue colored colonies on this. Now recombinants produce the colorless colonies. Now retrovirus. So these are viruses which will infect animals and they produce cancers in animals. Retroviruses can be disarmed to be used as a cloning vector, and they are made inactivated to make them as cloning vectors. Now competent host. Competent host is the host wherein bacterial cells will have the ability to take up the vector, and they contain the recombinant DNAs within them. Now methods to introduce this recombinant DNA to the competent host. How to do it? Cells are treated with divalent cations, which is Ca2 plus calcium ions, and these cells are incubated with recombinant DNA on ice, which is followed by heat shock technique at 42 degrees centigrade, and then they are put back into the ice. By this, the bacteria will be able to take up the recombinant DNA. Now coming to microinjection. So recombinant DNA is directly introduced into the nucleus of the cell animal with the help of the microinjections. Microinjections are very similar injections, but their needles will be very small. Now next is biolistics, which is the gene gun. The cells are bombarded with high velocity microparticles of gold and tungsten and disarmed vectors, as in case of A, tumefacients and retrovirus. Now processes of RDT, which is the isolation of gene material, that is DNA. For the process of RDT, DNA must be available in its purest form, and first of all, cells are supposed to be treated with specific chemicals to break open the cells and release the cellular components like DNA, RNA, proteins, etc. And contaminants like RNA and proteins are digested with the help of ribonucleases and proteases respectively, and addition of the chilled ethanol ultimately will precipitate out from the purified DNA, which can be seen as a collection of threads in the suspension. Now cutting of the DNA at specific locations. So DNA is cut into fragments with the help of restriction enzymes, and fragments which are generated after the restriction enzyme are isolated with the help of gel electrophoresis. And recombinant DNA is obtained by hybridizing gene of interest with vector, and this is done with the help of DNA ligase. Now polymerase chain reaction, PCR techniques. So recombinant DNA can be amplified by PCR. So several identical copies of it is synthesized in vitro, and two sets of primers which are chemically synthesized oligonucleotide sequences will stretch, and they are complementary to the region of the DNA. And then now these primer enzymes, which is the DNA polymerase and deoxynucleotides, are also added to it. Now PCR is of three techniques, which is denaturation, that is double helical DNA is denatured by providing high temperature and it is disintegrated. Now DNA polymerase will not get degraded in such high temperatures since it is used in this reaction in thermostatable, and it is isolated from the thermophilic bacteria, which is Thermus aquaticus, TAQ. Now next is the extension process, wherein replication of DNA will occur in vitro. Now this cycle is repeated several times to generate 1 billion identical copies of the DNA. So this is how it is done: denaturation, annealing and extension. Now next is the insertion of recombinant DNA into the competent cells. So insertion of recombinant into the host is done by several methods. So few of them are transformation in case of bacteria, and disarm vectors, biolistics and microinjections in case of plants and animal cells. And the cells bearing recombinant DNA will be selected because the recombinants will exclusively have selectable markers which are present in them. That is, they are similar to the antibiotic resistance. Now obtaining foreign gene products. So this is the stage for which the recombinant DNA was actually produced. So now the cell consisting of recombinant DNA will produce a novel protein product, which is desirable product, or the recombinant protein which is required for us. And for large scale production of the desirable products, that is the antibiotics, vaccines, enzymes, the optimum conditions are to be provided, and continuous culturing is to be done, wherein used culture media is drained from one side and fresh culture media is added to the other side. And cells are kept throughout in their log in an exponential phase. Now larger biomass is produced by this method, leading to the higher yield. And next is the bioreactors, wherein large vessels in which large volumes, that is 100 to 1000 liters of cultures are produced. Optimal growth conditions for microbes are present, like the temperature, pH, substrate, salts, vitamins, etc. So this is how they are produced. These are the bioreactors, wherein air is bubbled into the reactor and then it is sterilized first, and then the microbes are released for the growth. Now a bioreactor has following component agitators. So what is it? Agitator system, oxygen delivery system, foam control system, temperature and pH control system, sampling the ports. Now downstream processing. So what is it? It is a biosynthesis of many components like enzymes, alcohols, antibiotics, which take place within the bioreactor. Now the products so obtained are crude. So they are the raw materials and they require separation, purification and finishing, which is done under the downstream processing. Now DSP makes a crude bioproduct marketable, and after proper separation and purification, the preservatives are then added and the finished product is made to undergo clinical trials. And the quality checks is done before being sent to the market. So this is how the downstream processing will help in the proper notifications of these antibiotics or vaccines, etc. Thank you.